Cell biology. All living things are made up of cells. The cell as the basic unit of structure and function. These generalized statements are known as cell theory. This theory was forwarded by Matthias Slater and Theodor Schwann in 1838-39. Today, the cell theory includes four more ideas. The cell are the building block of structures and living things. The cell is derived from other cells by division. The cell contains information that is used instructions for growth, development, and functioning. The cell is the functioning the chemical reaction of life take place within cells. The ideas and concept of cell biology evolved during the 19th century as a result of gradual advancement in the field of microscopy and biochemistry. Today, study of the structure of cell cytology is part of major branch of biology known as cell biology. Due to its wide application, many new branches have sprung up in biology. Some of the new branches related to cytology are cytotaxonomy, cytogenetics, cell physiology, cytochemistry, molecular biology, cytopathology, cytoecology, microscopy. The cells are very minute and complex organizations. The small dimensions and transparent nature of cell and its organelles pose problems to cell biologists trying to understand its organization and functioning. Various instruments and techniques have been developed to study cell structure, molecular organization, and function. The diameters of majority of cell range from 5 to 500 micrometer, but most are between 10 to 150 micrometer. The systematic international system units of length are 1 meter, 1 millimeter, 1 meter, 1000 millimeter, 1 millimeter, 1000 micrometer, 1 micrometer, 1000 nanometer, 1 nanometer, 1000 picometer (pm). The angstrom is 10 minus 10 at sometimes used to record the thickness of cell membranes and the sizes of macromolecules while viewing objects. Human eyes have limited distinguishing or resolving power. The ability to reveal minute detail is expressed in terms of limit of resolution. It is the smallest distance that may separate two points on an object and still permit their observation as distinct separate points. The resolving power of naked human eye is 0.1 millimeter or 100 micrometer. It means that we cannot differentiate any points that are closer than this. Hence, we need instruments capable of high resolution to see smaller objects. Power of magnification. It is different from resolving power. Magnification is the increase in size of optical image over the size of the object being viewed. Increased magnification without improved resolution results only in a large blurred image. The human eye has no power of magnification. The first useful compound microscope was invented by Francis Johnson and Jacques Johnson in 1590. It has two lenses with magnification powers between 10x, 30x. Galileo Galilei 1564-1642 invented a simple microscope to study compound eye of insect. His microscope had only one magnifying lens. Marcello Malfigi 1628-1694, an Italian microanatomist, used a microscope to study organ tissues of animals. Robert Hooke, an English microphysicist, as 1665 examined a slice of cork tissue under a compound microscope built by him. He coined the term cells to honeycomb of cells in cork tissue. Enter one Leeuwenhoek improved the quality of lenses used in microscopes. His microscopes achieved magnification up to 300x. He was the first to observe free living cells. Further advancements in cell biology were made by improving the quality of compound microscope. Compound light microscope. This microscope was visible light for illuminating the object. It contains glass lenses that magnify the image of the object and focus the light on the retina of the observed eyes. It has two lenses, one at each end of a hollow tube. The lens closer to the object being wide is called objective lens. The lens closer to the eye is called ocular lens or eyepiece. The object is illuminated by light beneath it. A third lens called condenser lens is located between the object and the light source and it serves to focus the light on the object. Dark, dark field microscope. This type of microscope is useful for viewing suspension of bacteria. It is a special condenser that allows only rays of light scattered by structures within specimen. The result is an image that appears bright against a dark background with a high degree of contrast. The process is similar to seeing dust particles floating on a sunbeam. Phase contrast microscope. The phase contrast microscope has special fitments to the objective lens and substage condenser, the effect of which is to exaggerate the structural differences between the cell components. As a consequence, the structures within living unstained cells become visible in high contrast and with good resolution. Phase contrast microscopy avoids the need to kill cells or to dye to a specimen before it is observed microscopically. IL immersion microscopy. In early immersion microscopy, the light gathering properties of the objective lens are enhanced by placing IL in the space between the slide and objective lens. Normally, the technique is used to work permanently mounted specimens. The IL immersion lens gives higher magnification than the normal high for objective lens. Electron microscopy. The electron microscope uses the much shorter wavelengths of electrons to achieve resolutions as low as 3 angstrom. Electromagnetic coils are used to control and focus a beam of electrons accelerated from a heated metal wire by high voltages in the range of 20,000 to 1 lakh volts. The electrons are made to pass through the specimen electrons. They do passes out of the object are focused objective coil. Finally, magnified image is produced by a projector coil or lens. The final image on a screen or is recorded on photographic film to produce electron micrograph. This type of electron microscope is called transmission electron microscope. In a compound light microscope, the image is formed due to differences in light absorption. The electron microscope forms images as a result of differences in the way electrons are scattered by various regions of the object. The degree to which electrons are scattered is determined by the thickness and atomic density of the object. Hence, the specimens used in electron microscopy must be extremely thin. Living cells which are wet cannot be viewed in electron microscope. Scanning electron microscopy. This microscope has less resolution power than the TM, about 200 angstrom. However, it is very effective tool to study surface topography of a specimen. The whole specimen is scanned by a beam of electrons An image is created by electrons reflected from the surface of the specimen. Scanning electron microscope show depth of focus and three-dimensional image of the object. Cytological techniques. Cells are transparent and optically homogeneous organizations. They can be observed either directly or after preservation. For direct observation, the specimen needs sufficient contrast. Direct observation is possible by using vital stans. Vital stans. Vital dyes or stains are taken up by living cells without killing them. They selectively stain intracellular structure without affecting cellular metabolism and function. For example, genus green bee selectively stains mitochondria, Golgi apparatus, nuclear chromatin, and a dividing cell can be stained by methylene blue. Neutral red dye or Congo red dye can be used to stain yeast cells preserved in stained tissues. For detailed microscopic study, tissues containing cells are passed through various stages of cell preparation on a glass slide involves scaling, fixation, dehydration, embedding, sectioning, staining, and mounting. Killing and fixation. The process causes sudden death of cells or tissues and preserves freshly killed tissues in as lifelike a condition as possible. Affixative prevents bacterial decay and autolysis. It will also make different cell components more visible and prepare the cell for staining. The commonly used fixatives are acetic acid, formaldehyde, Bowen solution, and corneous fluid. Dehydration. In this process, water vapors are removed from cells or tissues using chemical agents. It is done by using ethanol and benzene. Embedding. The tissues are infiltrated with molten paraffin wax and hardens up on cooling and provides enough support to allow thin sections. Very thin sections need to be taken for electron microscopy. Hence, plastics are used for embedding. 4. Sectioning. The embedding material is cut into thin sections of needed thickness. It is done by using an instrument called microtome staining. The sections are immersed in dyes that stain some structures better than others. For example, cytoplasm stains pink with eosine, nucleus stains blue with hematoxylin, or red with saponin. Dehydration. Stained sections are immersed in ethanol to remove water. The tissue becomes more transparent. Dehydration is done gradually by using a series of increasing concentrations of ethanol in water. Finally, the section is placed in absolute alcohol. 7. Mounting. Clean sections are mounted on a side using a suitable medium like Canada balsam. A cover tip is added and the medium is allowed to dry. Ultrastructure of typical animal cell, centrioles, nuclear envelope, nucleus, nucleolus, smooth endoplasmic reticulum, Golgi apparatus, peroxisome, mitochondria, lysosomes, plasma membrane, rough endoplasmic reticulum, plasma membrane. Most important, it is the outer limiting membrane of both prokaryotic and eukaryotic cells. It is an ultra thin, elastic, living membrane. Plasma membrane is dynamic and selective transport barrier. Since the plasma membrane is ultra thin It could be observed only under electron microscope. Structure of the membrane is studied by isolating the same from the cell and conducting biochemical investigations. In 1895, Orton suggested that the membrane is made of fatty substances. Other workers later concluded that two layers of lipid were present in the cell membrane. According to model proposed by Danieli and Davison in 1935, the lipid bilayer of the membrane was coated on either side with protein. Protein coat, lipid bilayer, and protein coat. Danieli Dawson model of the cell membrane, old model. Robertson, using electromicrographs, proposed a unit membrane hypothesis. According to this hypothesis, the two outer layers of protein are about 2 nanometers thick and appear densely granular, then close a clear central area of about 3.5 nanometer wide, consisting of lipids. The lipids are mainly phospholipid molecule, glycoprotein, glycolipids, phospholipids, cell count, cholesterol, lipoprotein bilayer, sphingolipids, the fluid mosaic model of cell membrane, most important model for papers. Singer and Nicholson have proposed a fluid mosaic model for the plasma membrane. The fluid mosaic membrane is a dynamic structure. In this structure, much of the protein molecules float about. Some of them are anchored to the organelles within the cell. Lipid molecules are move about. Fluid mosaic model is applied to all biological membranes in general. The cell membrane controls the passage of material both into and out of the cell. It regulates the passage of water and dissolved substances. Water passes through the membrane by osmosis. Water soluble substances cross the membrane by diffusion or by active transport. Many water soluble solutes are transported by carrier protein. Lipid soluble compounds pass more quickly by dissolving in the phospholipid layer. Most important topic, mitochondria, especially for examination. The mitochondria are filamentous or granular cytoplasmic organelles of all aerobic cells of higher animals and plants. They are also found in microorganisms, including algae, protozoa, and fungi. They were first observed by Kolliker in 1850 as granular structures in the muscles. The name mitochondria was given to them by Benda. Various steps up glycolysis in mitochondria was discovered by two German biochemists, Anden and Marhop. Emden got the Nobel Prize in 1922. Sir Hans Adolf Krebs in 1937 found out various reactions of citric acid cycle. Kennedy and Langer 1948 to 50 showed that citric acid cycle oxidative phosphorylation and fatty acid oxidation took place in the mitochondria. The number of mitochondria in a cell depends on the type and function state of the cell. Certain cells contain large number of mitochondria. For example, sea urchin contain 1,40,000 to 1,50,000 mitochondria. Oocytes of amphibians contain 3 lakh mitochondria. Liver cells of rat contain only 500 to 1600 mitochondria. Some algal cells may contain only 1 mitochondria. The mitochondria may be filamentous or granular in shape. They vary in size from 0.5 micrometer to 2.0 micrometer. Due to their minute nature, they cannot be seen under light microscope. Each mitochondrion is bound by two highly specialized membranes. The outer membrane is smooth. It is separated from the inner membrane by 6 to 8 nanometer wide space. The inner membrane is highly convoluted, forming series of unfolding known as cristae. Three-dimensional structure of mitochondria matrix, interstitial space, cristae, inner membrane, outer membrane, interstitial space, oxysomes, crystal. Thus, mitochondria are double membrane envelopes. The inner membrane divides the mitochondrial space into two distinct chambers. The outer compartment is perimitochondrial space. It is found between outer and inner membrane. The inner compartment is the matrix space. It is filled with a dense gel-like substance called mitochondrial matrix. The matrix contains lipids, proteins, and circular DNA molecules. The outer and inner membranes, intermembrane space, and mitochondrial matrix contain several enzymes. Hence, the mitochondria perform several important functions such as oxidation, dehydrogenation, oxidative phosphorylation, and respiratory chain of the cell. Since mitochondria play a key role in oxidation of carbohydrates and fats, they are considered as the actual respiratory organ of the cell. During such biological oxidation, large amount of energy is released. The energy is utilized by the mitochondria for synthesis of energy rich compound known as adenosine triphosphate or ATP. Due to this function, the mitochondria are also known as powerhouses of the cell. An animal cell's mitochondria produce 95% of ATP molecules. Ribosomes. The ribosomes are small, dense, rounded, and granular particles. They contain ribonucleoprotein. They freely in the matrix of the mitochondria, chloroplast, and cytoplasm are remain attached with the membrane of the cytoplasmic reticulum and nucleus.